Okay, let's unpack this. When you think about where, like, the basic building blocks of life come from, you might think geology, maybe the carbon cycle in the air.
Hmm.
But there's this really critical link between the raw stuff in the Earth's crust and, well, everything growing above ground. And that's plants.
Absolutely. They're kind of the original miners,
Yes.
aren't they? Pulling those essential inorganic elements out of the soil and bringing them into the whole living system, the biosphere. They really form the base.
Right. And studying how they do that, how they acquire and use these elements, that's mineral nutrition.
That's the field, yes.
Yeah.
It gets pretty deep into the science, chemistry, biology,
Mm-hmm.
but it's also incredibly practical.
Practical how, exactly?
Well, it's fundamental to feeding the world, really.
Mm.
Modern high-yield agriculture just wouldn't work without providing plants the right mineral nutrients, usually fertilizers.
Hmm.
If you look at figure 5.1 in the materials, you can see global fertilizer use just nitrogen, phosphorus, potassium. It's just exploded. Like 30 million metric tons in 1960, up to around 180 million recently.
Wow, 180 million tons. That's a six-fold jump.
Yeah.
That scale must have some major ripple effects, good and bad.
Exactly. Good side. Massive increase in food production.
Yeah.
Absolutely vital. But the costs are significant, too.
Like what?
Well, energy, for one. Making nitrogen fertilizer uses more than half of all the energy consumed in agriculture. Phosphorus,
Yeah.
that comes from mines, it's a finite resource.
Okay.
And plants often only take up, a fraction of what's applied. The rest, it can run off into rivers and lakes, causing pollution, think nitrate in drinking water wells. Or it contributes to atmospheric nitrogen deposition, which is actually changing whole ecosystems.
So it's this constant balancing act, production versus environment. Though you mentioned plants can be part of the solution sometimes.
Yeah, I mean, traditionally, manure recycling puts nutrients back. And now there's phytoremediation, using specific plants to actually clean up contaminated soils by sucking up heavy metals and things.
Huh, interesting. Using plants as cleanup crews.
Sort of, yeah.
Okay, so plant mineral nutrition. It's about this core process of getting elements, but it connects to agriculture, energy, pollution, even cleanup. So today, we're diving into a chapter that really breaks this down. What nutrients plants need, how we know if they're missing them,
Right, the deficiency symptoms.
the whole soil environment, how roots work down there, and these amazing partnerships they form with microbes.
It really shows you the incredible ways plants have adapted just to get the basic stuff they need to build themselves.
So let's dig in together and see what's going on beneath the surface. All right, first things first. How do scientists even know which elements a plant absolutely has to have? What makes something essential?
Yeah, there's a pretty strict definition for that. An element is essential if, one, it's a core part of the plant structure or is needed for a specific metabolic job.
Oh, okay.
And, crucially, if the plant can't complete its whole life cycle, germination, growth, reproduction, making viable seeds without it. If a plant have all the essential elements, plus, water, CO2, light, it can make everything else it needs.
So elements like carbon, hydrogen, oxygen, they make up most of the plant's weight, but they come from air and water. So they aren't usually called mineral nutrients in this context.
Correct. Mineral nutrients are the ones that typically come from the soil as ions.
And we often hear them split into two groups based on how much the plant needs: macronutrients and micronutrients.
Yes, that's a common way to categorize them based on their concentration in the plant tissues. You can see that in Table 5.1. Macros are needed in larger amounts, say 0.1% of dry weight or more. Micros are needed in tiny amounts, parts per million level.
But the notes say the division isn't always perfect physiologically.
Exactly. It's really just about quantity. Some micronutrients are just as critical for survival as the macronutrients. The plant just needs way less of them, like vitamins for us, right?
Yeah.
Tiny amounts, but essential.
Okay, so maybe a better way to think about them is what they do, their function.
That can be really helpful, yeah. Table 5.2 actually groups them that way, by biochemical function. You've got group 1, elements that become part of carbon compounds like nitrogen and sulfur,
Right.
often involved in redox reactions.
Right, nitrogen and proteins, chlorophyll.
Exactly. Then group 2, elements involved in energy storage or structure. Think phosphorus and ATP in cell membranes, or silicon and boron, sometimes in cell walls.
Okay, energy and structure.
Group 3 are elements that mostly stay as ions. Things like potassium, calcium, magnesium. They act as enzyme cofactors, or they're really important for osmotic balance, controlling membranes.
So regulation, keeping things running smoothly, and the last group.
Group 4 involves elements primarily involved in redox reactions, electron transfer. Iron, manganese, copper, molybdenum, key parts of enzymes and electron carriers in things like photosynthesis and respiration.
That functional grouping really helps understand why they're essential. So how did researchers figure all this out? You can't just easily remove only potassium from soil, can you?
No, that was the big challenge. The breakthrough really came back in the 19th century with solution culture, hydroponics.
Ah, growing plants in water.
Right. Researchers like Sachs and Knott Prue's plants don't need the soil material itself. They just need the essential mineral ions dissolved in water, plus light and air, of course.
That must have completely changed how they studied nutrition.
Absolutely. Hydroponics lets you control the nutrient supply precisely. Add this element, leave out that one, see what happens. It's still a vital tool for research, and it's used commercially, too, in various forms you see in Figure 5.2.
But keeping those solutions balanced must be tricky. The notes mention iron being a problem.
Yes, iron is notorious. It precipitates out of solution very easily at neutral or higher pH, becoming unavailable. That's where chelates come in.
Chelators.
Yeah, compounds like EDTA, you can see its structure in Figure 5.3. They basically grab onto the iron ion like a claw and keep it dissolved and available for the roots to take up. Same for some other medications, too.
Huh. Clever chemistry to solve a biological problem. Okay, so using controlled setups like hydroponics, you figure out what plants need. But what happens out in the real world in soil when a plant doesn't get enough of something? That's when you see deficiency symptoms.
Precisely. And these symptoms are basically the outward expression of some internal metabolic mess-up caused by the lack of that specific element. Now, diagnosing these in soil is harder than in hydroponics.
Why is that?
Well, you might have multiple deficiencies at once, or maybe a soil problem like poor drainage, or even a disease that looks kind of similar. It gets complicated.
But the chapter gives us a really key diagnostic clue: how mobile the element is inside the plant. Table 5.4 shows this.
Yes, this is crucial. Some elements like nitrogen, phosphorus, potassium, magnesium, they're mobile.
Yeah.
The plant can actually move them from older tissues to the newer, actively growing parts like young leaves and buds.
So if one of those is lacking,
the symptoms show up first on the older, lower leaves. The plant is essentially scavenging the nutrient from the old parts to feed the new growth. It sacrifices the old for the young.
Got it. Old leaves look bad. Think mobile element deficiency. Makes sense.
Right. Then you have the immobile elements: calcium, sulfur, iron, boron, copper. Once these are built into tissues, they're pretty much stuck there. The plant can't easily move them around.
So for those, the symptoms appear where?
On the younger leaves, or the growing tips, the meristems. Because the new growth can't get a supply shipped in from the older parts, it suffers first.
Younger leaves or tips look bad. Think immobile element. Okay, that's a super helpful rule of thumb. Let's walk through a couple of examples. Nitrogen mobile, you said?
Right. Needed in huge amounts. Proteins, chlorophyll, nucleic acids. Deficiency leads to stunted growth and that classic overall yellowing chlorosis started on the older leaves. They turn pale green, then yellow, maybe tan, and can drop off. Younger leaves stay green longer.
Okay, fits the mobile pattern perfectly. How about phosphorus? Also mobile.
Yep. Key for energy, ATP, membranes, DNA. Deficiency also causes stunting, but often the leaves turn a dark, kind of bluish green. Sometimes you see purple patches, especially on the underside from anthocyanin pigments building up, and maybe some dead spots, necrosis. Again, shows up on older leaves first.
Dark green and purple on old leaves. Different look, but same mobile location. Okay, now an immobile one. Calcium.
Calcium's big for cell walls, membranes, and also signaling inside the cell. Since it's immobile, deficiency hits the young, actively growing parts hard. You see necrosis, tissue death, right at the root tips or the shoot tips, the young emerging leaves. Young leaves might look deformed, maybe hooked at the tip.
Death at the tips. Deformed new growth. Clear immobile pattern. And iron? Also immobile.
Iron is critical for enzymes involved in electron transport, like in chlorophyll synthesis. Deficiency causes intravenous sclerosis.
Meaning?
The tissue between the leaf veins turns yellow or pale, but the veins themselves stay green, at least initially. And because iron is immobile, this pattern shows up first on the younger leaves.
So yellow between the veins on young leaves is iron. But if it was on old leaves, you might think magnesium, which is mobile.
Exactly. That comparison really highlights how useful mobility is for diagnosis. Location, location, location.
So looking at where symptoms are and what they look like gives you big clues. how do growers actually check nutrient status in practice?
Well, there's soil analysis. Testing the soil itself gives you an idea of the potential supply of nutrients. But soil's complex, availability changes, so it's not the whole picture.
Right.
More direct is plant tissue analysis.
Analyzing the plant itself.
Yeah, you take leaf samples, measure the nutrient concentrations in them. This tells you what the plant has actually managed to take up. Then you compare those levels to established standards, like in Figure 5.4. There's a deficiency zone, where more nutrient means more growth. Then an adequate zone where the plant has enough and adding more doesn't help yield. And then potentially a toxic zone if levels get too high. The goal is to keep levels in that adequate zone, often guided by the critical concentration, the minimum needed for maximum growth.
So tissue testing helps fine-tune fertilizer use, avoid waste and potential toxicity.
Precisely. It tells you what the plant is actually experiencing, nutrient-wise.
Okay, so you've diagnosed the deficiency using symptoms, mobility clues, maybe tissue tests. How do you fix it?
Well, in modern farming, the main way is adding fertilizers. Historically, farming involved a lot more nutrient recycling, animal manure, crop residues going back onto the fields.
Right.
But high production systems today remove a lot of nutrients in the harvested crops, so you generally have to replace them.
And fertilizers are basically chemical or organic.
Broadly, yes. Chemical fertilizers are inorganic salts, usually providing the big three, nitrogen and phosphorus P and potassium K. Sometimes specific single nutrients, sometimes blended compounds. Organic fertilizers are things like manure, compost, bone meal, natural materials.
Does the plant care where the nutrient comes from?
Fundamentally, no. Plants absorb nutrients primarily as inorganic ions, nitrate, ammonium, phosphate, potassium ions, etc., whether that ion comes directly from a dissolved chemical salt or from microbes breaking down organic matter. The plant takes it up in the same form. Organic fertilizers do have other benefits, though they release nutrients more slowly and improve soil structure.
Besides adding fertilizer, you mentioned managing soil pH is important. Figure 5.5 really shows how much pH affects availability.
Oh, massively. pH controls the chemical form of nutrients and how tightly they bind to soil particles. Most nutrients are best available in slightly acidic soil, maybe pH 5.5 to 6.5. If soil is too acidic, you might add lime, calcium carbonate, to raise the pH. If it's too alkaline, adding sulfur can lower it. Sometimes adjusting pH is enough to unlock nutrients already in the soil.
And sometimes you can just bypass the soil altogether. Foliar feeding.
Exactly. Spraying nutrient solutions directly onto the leaves. It's a way to get nutrients in quickly, especially if soil conditions are bad. Maybe the soil locks up iron really tightly or the roots are damaged.
How does that work? Do they just soak through the leaf?
They diffuse through the leaf cuticle, the waxy outer layer, and also can enter through the stomata, the small pores. You often add surfactants, like detergents, to help the solution spread out in a thin film. It's particularly useful for micronutrients or for a quick fix.
So add to the soil, tweak the soil pH, or spray the leaves different tools for the job. Okay, let's shift focus now to where all this action happens. The soil itself. It's way more than just dirt, isn't it?
Oh, absolutely. Soil is this incredibly complex mix. You've got the solid phase mineral particles from weathered rock, plus organic matter from decaying stuff. That's the source of many nutrients. Then there's the liquid phase, the soil solution, water with dissolved ions, which is how nutrients move around, and the gas phase air, filling the pore spaces, crucial for root respiration.
And it's teeming with life, too.
Hugely. Roots themselves, obviously, but also bacteria, fungi, insects, worms, a whole ecosystem. Microbes are especially key. They break down organic matter, releasing nutrients in a process called mineralization. But they can also compete with roots for those same nutrients or, as we'll see, form vital partnerships.
One key chemical feature is that many soil particles, especially clay and organic matter, have negative charges on their surfaces.
That's right. And that negative charge is really important because it attracts and holds on to positively charged ion stations. Things like potassium, K+, calcium, Ca2+, magnesium, Mg2+, ammonium, NH4+.
So they stick to the soil.
They absorb onto the surface, yeah. This is actually good because it prevents them from being easily washed away, leached out by rainwater. It creates a nutrient reservoir.
But if they're stuck, how do plants get them?
They're not permanently stuck. They can be swapped out, exchanged with other cations in the soil solution, including hydrogen ions that roots release. This is called cation exchange. You can see it illustrated in Figure 5.6.
Okay.
The soil's capacity to hold these exchangeable cations is its cation exchange capacity, or CEC. Table 5.5 shows some typical values. Soils with more clay or organic matter have a higher CEC, meaning a larger nutrient reserve.
What about the negatively charged nutrients, the anions, like nitrate, NO3?
Well, since charges repel, these anions aren't held by the negatively charged soil particles. They tend to stay dissolved in the soil water. This makes them much more mobile.
And much more likely to get washed away.
Exactly. Leaching is a much bigger issue for anions like nitrate and chloride.
But you mentioned phosphate earlier. That's an anion, PO43. But it's known for being really immobile in soil. Why?
Phosphate is kind of a special case. It reacts very strongly with iron and aluminum compounds in acidic soils and with calcium in alkaline soils, forming highly insoluble precipitates. You can see this pH effect in figure 5.5 again. It gets locked up chemically, not just held by charge, which severely limits its movement and availability.
So getting enough phosphate is often a challenge for plants.
The major one, yes.
Okay, so the soil itself, its chemistry, its pH, it sets the stage. How do roots actually navigate this and get what they need?
Well, first off, plants invest heavily in roots. The sheer scale can be mind-blowing. The chapter mentions a study on a single winter rye plant. After just 4 months, its root system was estimated to have a total length of over 500 kilometers.
500 kilometers from one plant.
And the total surface area, including all the tiny root hairs, was estimated at something like 500 square meters.
Wow, that's huge, like half a tennis court of surface area underground.
Exactly. It's all about maximizing contact with the soil volume to absorb water and nutrients. And roots don't just grow randomly. They can sense where nutrients are richer and actually grow more prolifically in those patches. Figure 5.11 shows that.
Smart roots. And they're always growing, pushing into fresh soil, right? That happens at the tip.
Yes. The root tip is key. Figure 5.9 shows the zones. You have the apical meristem, where cells divide, protected by the root cap. The cap also senses gravity and secretes mucilage, a slimy substance that lubricates the passage through soil.
And behind that,
The elongation zone, where cells expand, pushing the tip forward. This is also where the endodermis develops, with that important Casparian strip.
yes, the gatekeeper layer.
Right. It's a waxy band in the cell walls that forces water and solutes to cross a cell membrane to enter the vascular tissue, the xylem. It gives the plant control over uptake. Further back is the maturation zone, where root hairs develop.
Root hairs, they must massively increase the surface area.
Dramatically. Especially important for absorbing those curly mobile nutrients like phosphate, because they explore the soil immediately around the main root very thoroughly.
So once a root is in place, how do the nutrients actually move from the soil to the root surface? They don't just teleport in.
2 main physical processes are involved: bulk flow and diffusion.
Okay, what's bulk flow?
That's simply nutrients being carried along with the water that's flowing towards and into the root. This water movement is driven primarily by transpiration. Water evaporating from the leaves pulls water up through the plant. Mobile nutrients like nitrate that are dissolved in the soil water get swept along for the ride.
So they hitch a ride on the water current. And diffusion.
Diffusion happens because roots are actively absorbing nutrients right at their surface. This lowers the concentration of that nutrient in the soil solution immediately next to the root. So if the concentration is higher further away, the nutrient will tend to move or diffuse down its concentration gradient towards the root surface.
And this is more important for...
For the less mobile nutrients like phosphate or potassium that don't move much with bulk flow, the root creates a sink and diffusion slowly refills the area around it.
But if the root takes up nutrients faster than diffusion or bulk flow, can resupply that area right next to it.
Then you get a nutrient depletion zone, exactly as shown in figure 5.10. A region around the root where the concentration of that nutrient is significantly lower than in the bulk soil further away.
Which really emphasizes why roots need to keep growing, right? To escape their own depletion zones and explore fresh soil.
Absolutely. Continuous growth is key, especially for immobile nutrients.
Which perfectly sets up the final piece of this puzzle, those microbial partnerships you mentioned earlier. The plant isn't always working alone down there.
Not at all. That hidden world of plant microbe interactions is incredibly important. And the prime example is mycorrhizal symbiosis.
Mycorrhizae, fungus roots.
Exactly. Associations between plant roots and specific types of soil fungi. And this isn't some rare exception, it's a norm. Something like 90% of all land plant families form mycorrhizae, including most of our crops.
What's the deal in this partnership?
It's a classic symbiosis, a mutual benefit. The plant provides the fungus with sugars, carbohydrates produced during photosynthesis. The fungus, in return, helps the plant acquire nutrients, especially phosphorus and sometimes nitrogen, and can also improve water uptake and even protect against some root diseases.
How does the fungus help get nutrients?
The fungal hyphae, these microscopic filaments, extend way out into the soil, far beyond where the root hairs reach. They act like a massive extension of the root system, exploring a much larger volume of soil.
So they can access nutrients the root itself can't reach, particularly the immobile ones.
Precisely. They're incredibly good at scavenging phosphorus, reaching beyond that depletion zone around the root. They can also tap into forms of nutrients the plant can't use directly sometimes.
The chapter mentions two main types of mycorrhiza.
Yes. The most common type found in the vast majority of plants, including most crops, are the arbuscular mycorrhizas, or AM.
Arbuscular.
It refers to arbuscules, these highly branched tree-like structures the fungus forms inside the root cortex cells. You can see them in figure 5.13. This is the site where the nutrient exchange happens between fungus and plant. AM fungi are superstars at phosphorus uptake.
Okay, AM inside the cells, great for P. What's the other type?
Ectomycorrhizas, or ECM. These are less common overall, mostly found on trees and shrubs like pines, oaks, birches. Ecto means outside them. These fungi don't usually penetrate into the root cells.
So how do they work?
They form a thick sheath or mantle of hyphae around the root tip and then grow extensively between the outer root cells, forming a network called the Harding net, figure 5.14. Their hyphae also explore the soil widely, like in figure 5.5.
And what are they particularly good at?
ECM fungi are really important for accessing nutrients tied up in organic matter. They can produce enzymes that break down organic molecules to release nitrogen and phosphorus, which is crucial for trees growing in forest soils, where much of the N and P is in organic forms, things non-mycorrhizal roots struggle to get.
So AM are intracellular specialists for mineral P, while ECM are extracellular specialists, especially for organic N and P in trees.
That's a good way to summarize the key difference in their nutrient acquisition strategies. The actual transfer across the fungus-plant interface is complex, involving specialized transporters on both sides.
Seems like these fungi are incredibly beneficial allies for plants.
Hugely beneficial,
Yeah.
which makes it concerning that the material points out some common agricultural practices can actually harm them.
Like what?
Intensive tillage, which breaks up the hyphal networks in the soil, high levels of fertilizer application, especially phosphorus, which can make the plant less interested in supporting the fungus, and soil fumigation, which can kill them off.
So in some of our modern farm fields, the plants might be missing their fungal partners.
It's often the case. The non-mycorrhizal state we see in many crops today might actually be, as the text puts it, an artifact of these practices, rather than the plant's natural state.
Which definitely makes you think about whether we could be doing things differently to leverage these natural systems better.
Wow. Okay, that was quite a journey. We went from the basic definition of essential elements, figuring out what plants need, and how to spot deficiencies using symptoms and mobility,
Right, the old leaves versus young leaves distinction.
to the really complex world of the soil itself, the
Right.
charged particles, the pH challenges, how nutrients like phosphate get locked up.
And how roots, with their incredible growth and structure, try to overcome that. The bulk flow, the diffusion, those depletion zones.
And finally, realizing that roots often don't work alone, relying on these vital mycorrhizal fungi to extend their reach and access nutrients, especially phosphorus and organic forms. It really paints a picture of plants as incredibly sophisticated systems, constantly interacting chemically and biologically with their environment just to get the raw materials they need to grow. And understanding all this is just fundamental if we want to feed everyone sustainably and look after the environment.
Absolutely. That hidden world right beneath our feet is just unbelievably dynamic and complex.
So think about that next time you're looking at a plant.
Hmm.
Consider that vast, invisible network of roots and fungi working together underground. What if we could really understand and maybe even harness those natural partnerships more effectively? How might that change the future of agriculture and how we think about plant health?